information flows from DNA to RNA to proteins. That genetic code in DNA specifies the protein end product. So during transcription, which is what we're going to be talking about, that information is copied from DNA to RNA. DNA is the one with two strands, and RNA is single-stranded. But transcription later serves as the template for protein synthesis. So we're going to start off by talking about prokaryotic transcription. So it starts with RNA polymerase when it binds to a promoter sequence upstream of the gene. And bacteria proteins contain two key regions. So those regions are the -10 prib now box, TATAAT, and the -35 region, TTGACA. The sigma subunit recognizes these sequences and helps start the transcription. So next is the chain elongation, which is when the double helix unwinds the transcription start site. This exposes the template strand. Then the RNA polymerase moves along it, building RNA in the 5' to 3' direction. It's very important that it always goes 5' to 3'. And it uses complementary ribonucleotides. And then after the first few nucleotides, the sigma dissociates, as we see in the picture, and the elongation continues by the core enzyme. Okay, last step, termination. The transcription ends when the polymerase reaches the termination sequence. The RNA transcript may form a Harkin loop, like this one, or growth factor, like in this picture, which is an RNA helicase that reaches the RNA from the DNA template. In eukaryotes, transcription happens in the nucleus using RNA polymerase II. It requires many transcription factors and TATA binding proteins at the core promoter. Enhancers and silencers can increase and decrease transcription's efficiencies. So let's get more into the genetic code. We'll be starting to talk about mRNA processing. So the initial transcript is pre-mRNA. It undergoes modifications. A 5' cap is added, a poly A tail attaches to the 3' end, and introns are removed by spliceosomes. Through alternative splicing, one gene can produce several mRNAs. Now for the genetic code. The mature mRNA carries information as triplets of bases called codons. Each codon specifies one amino acid. These codons together form genetic code, which directs how proteins are built from the RNA message. Also, I just really love this picture because it shows how when the RNA comes in, the tRNA comes to the ribosome and it codes for the proteins, which make us, so it's just a really cool process. So overall, we see how important transcription is in our genetic code. We need it to transcribe the genetic material so that we can then translate and then get proteins that make us.